Luminosa, remarkably simple, precisely yours. Luminosa's InstaFLIM feature enables fast fluorescence lifetime image analysis with minimal user interaction. Fluorescence lifetime imaging provides new insights into various research areas such as cellular, developmental, or plant biology. By offering new experimental capabilities like multiplexing many fluorescent markers, characterizing tissue autofluorescence, removing autofluorescence that spectrally overlaps with marker signals, or sensing changes in the cellular environment. Until now, adopting fluorescence lifetime imaging required a steep learning curve because many parameters had to be set manually during a multi-step analysis process. Moreover, there are different analysis routines to choose from. So far, researchers had to run them separately and gather results afterwards to get a comprehensive picture. With Luminosa's new software, a fast GPU-based algorithm delivers all results within a few seconds. 4 different analysis routines run in parallel. A pixel-by-pixel multi-exponential decay fit with up to 5 lifetimes, from which a suitable fit model is automatically selected, and the corresponding overlay image created. A phasor plot is calculated, in which regions of interest can be selected to locate the associated structures in the image. In the lifetime histogram, certain lifetime ranges can also be chosen to highlight the corresponding structures in the image. In this example, both the triple exponential fit and the phasor plot clearly separate tubulin and mitochondria. A triple species pattern matching analysis additionally identifies the DAPI nuclear stain. In conclusion, the InstaFLIM feature links and presents results from FastFLIM, pattern matching, phasor plot, and lifetime fitting next to each other, so that you can get a comprehensive picture of your sample, in this example for reliable species separation. Save time and simply focus on your samples. Use FLIM reliably and get results quickly with minimal user input. Luminosa, remarkably simple, precisely yours. Luminosa, remarkably simple, precisely yours. Luminosa's InstaFCS feature performs online FCS fitting, including standard deviation of the correlation curve. The software automatically suggests the best fitting model and parameters. Fluorescence correlation spectroscopy techniques provide information about molecular concentrations and diffusion coefficients. These are fundamental for studying, for example, liquid-liquid phase separation or biological membranes. Furthermore, cross-correlation reliably reveals binding and co-localization. So far, acquiring good FCS data was not trivial because many factors need to be taken into account. The sample has to have the right concentration. The size of the labeled molecule and the viscosity of the sample need to be considered. Furthermore, the excitation power and triplet state behavior influence the correlation curve as well as the size and shape of the observation volume. Important acquisition parameters, such as the excitation laser power, had to be calibrated and set manually. After getting the data, the next challenge was to select the proper fitting model. Different models could be tried one after the other while manually adjusting parameters. In Luminosa's new software, selection of the employed fluorophores automatically loads the appropriate acquisition parameters and analysis channels. Once enough photons are acquired after the start of the measurement, auto and cross-correlation curves and their standard deviation are calculated. A fast, GPU-based algorithm performs online fitting of the data with 4 models in parallel, which include pure diffusion of 1 or 2 species and a triplet state. It then automatically selects the best fitting models and highlights them with stars. For each, you can read out the concentrations and diffusion speeds live. This enables you to instantly evaluate the sample and data quality. If necessary, you can adjust acquisition parameters right away, saving valuable time. Save time and simply focus on your samples. Use FCS reliably and get results quickly with minimal user input. Luminosa, remarkably simple, precisely yours. Luminosa, remarkably simple, precisely yours. Luminosa's FRETcompass is a set of features that makes quantitative single-molecule FRET measurements more accessible. It primarily displays the FRET efficiency versus stoichiometry histogram live during the measurement. Moreover, it automatically determines correction factors from the data and uses them to correct the histogram in real time. The procedure is based on the community-driven benchmark study published in Nature Methods in 2018. Single-molecule FRET studies have become a standard tool for studying dynamic structural changes in proteins and nucleic acids. They can reveal dynamic events on timescales covering several orders of magnitude, from nanoseconds to several seconds. This allows studying, for example, folding, binding, aggregation, and allosteric signaling. The power of single-molecule FRET is highlighted by its ability to unravel intrinsically disordered proteins, whose biological relevance has been increasingly recognized in recent years. So far, single-molecule FRET experiments involved a lot of training, as acquiring a good single-molecule FRET dataset was a laborious process. Additional control measurements were necessary to manually determine correction factors for a precise analysis, a procedure which demanded a lot of instrumentation expertise. With Luminosa's new software, you simply select the FRET pair present in your sample, then it automatically loads the appropriate acquisition parameters and analysis channels. Based on the assigned channels, the software displays the efficiency versus stoichiometry histogram live as the data is collected. From the raw data, it automatically determines the correction factors once enough events are detected and uses them to correct the histogram in real time. The procedure is based on the community benchmark study published in 2018. The online preview enables you to instantly evaluate the sample and data quality. If necessary, you can adjust acquisition parameters right away, saving valuable time. You can dive even deeper into the data. For the first time, you can directly explore the fluorescence lifetime decays of the donor only, low FRET and high FRET populations in all different channels. Save time and simply focus on your samples. Confidently incorporate quantitative single-molecule FRET measurements into your daily routine. Luminosa, remarkably simple, precisely yours. Luminosa, remarkably simple, precisely yours. Luminosa's LumiFinder automatically detects and measures immobilized emitters. Single-molecule studies, in particular single-molecule FRET, have become a standard tool for studying dynamic structural changes in proteins and nucleic acids. The FRET effect acts as a molecular ruler that reports distances between 2 and 8 nanometers. Thus, it can reveal dynamic events on timescales covering several orders of magnitude from nanoseconds to several seconds. This allows the studying of, for example, folding, binding, substrate uptake, and allosteric signaling. The power of single-molecule FRET is highlighted by its ability to unravel intrinsically disordered proteins, whose biological relevance has been increasingly recognized in recent years. So far, researchers wishing to observe single molecules immobilized on a surface with a confocal microscope had to manually select each individual molecule in an image for further measurements, a very repetitive and time-consuming task. Many molecules need to be recorded to obtain enough statistics for a reliable analysis. Some labs developed their own custom software solution for data acquisition, usually relying on custom-built confocal microscope setups, but not every researcher has that possibility. The LumiFinder is supported by several of Luminosa's other features: a hold focus option for long acquisitions, laser power calibration for reproducibility, and pulsed interleaved excitation that controls for the presence of the acceptor in single-molecule FRET. Prior assignment of analysis channels enables online previews. An overview image of single immobilized FRET pairs is acquired. In the image, single molecules can be selected based on their intensity, FRET efficiency, or stoichiometry, and are automatically located by the LumiFinder. Changing a selection criterion, such as increasing the minimum intensity threshold, instantly updates the localizations. Automated point measurements are taken at the selected locations one by one. The online preview shows the intensity time traces of donor emission after donor excitation, of the FRET signal after donor excitation, and of acceptor emission after direct acceptor excitation. You even get the lifetime information for each trace. In order to get good statistics for precise analysis results, one has to record many molecules. To get such large numbers effortlessly, the LumiFinder can be applied to large sample areas with tiling and stitching. These long acquisitions are enabled by the hold focus option. Luminosa brings additional benefits to single-molecule imaging. You get the lifetime information for each molecule. The system offers increased specificity and lower background, and you can use it for studying both immobilized and diffusing molecules. Save time and simply focus on your samples. Take the next step. Reliably detect immobilized molecules with a confocal and efficiently acquire high-quality data sets that include lifetimes. Luminosa, remarkably simple, precisely yours.